focused on machine learning, AI, and topics on the agenda. Avoid spamming the chat, right? During the session, feel free to ask questions during the Q&A segment, and just have fun and enjoy the session while connecting with others. So welcome everyone. Our topic for today will be in genomics ML accuracy, not the same as equity, part 2. And our speaker is going to be Chioma. So a little bit about Chioma. Chioma Oselu Oyindo, I'm sorry if I pronounce her name wrongly, is a computational biologist who enjoys building both tools and communities. Her work centers on population genetics, where she focuses on developing prediction tools that work well for underrepresented populations, especially African cohorts. She contributes to open source projects like Bioconductor, BugSigDB, co-leads global training programs, and shares beginner-friendly bioinformatics resources on LinkedIn. All right. Okay. Give me a second, guys. All right. It seems that we're having some difficulty reaching her. She's going to be, or she may have be having some technical difficulties. So let's give her some time to join back into the call. Chioma, are you there? Okay. So it seems our host is having a bit of some technical challenges, right? So we're just going to, keep everyone active on the call by doing some icebreakers. So um can someone maybe take the floor and talk about something they've been working on in the AI space or something interesting that they've been, researching? Anyone, just raise your hand and I'll pick you. Are you guys shy? No one is, taking the floor. Anything at all that has interested you recently in the AI space and you'd like to talk about? Okay. Well, it seems I'm the only one speaking. So maybe I'll go ahead and everyone else can talk too. So maybe before I start, I just want to talk about for a second the Machine Learning Lagos grad school series. So post the link in the chat. Please register if you haven't already. If you were here for the last years or you know how very informative everything was, And it's also on YouTube. The 2025 grad school series is also on YouTube. It's on YouTube on our channel, and you can review that and get some expectations for what's going to happen this year. So I've posted the link on the chats. So go ahead and register if you haven't already. Hello, everyone. So to those who just joined the call, our speaker is experiencing some technical issues, and she will be back very soon. But before that, I asked everyone if they would be interested in talking about something in the AI space they recently found out or currently working on or researching. And they left me out hungry. So I'm going to start. Okay. So recently I've been looking up reinforcement learning. Who here has done anything on reinforcement learning? Anyone? And so far so good. It's been an interesting experience. You know how normal machine learning or classical machine learning deals with predicting a set value, right? Classification and regression, training on label data, and come to find out that something like reinforcement learning, where all right, it seems our speaker says she'll be joining us in 5 minutes. So just sit tight, everyone, and she will be with us very soon. Thank you. So I know we've all been anticipating this for a while. I mean, throughout the month of May. So yeah. So yeah, like I was talking about in reinforcement learning, is there anyone who has heard of it or or done any work in reinforcement learning? Because recently I've been exploring it and it seems really interesting. And so I also watched this really nice documentary about Google DeepMind and how they are focused on reinforcement learning. And yeah, it was a really cool documentary to watch and see how far they've gone from building reinforcement learning algorithms to beat master chess players in the world. I can drop the link to that documentary if anyone's interested. But, okay, I would definitely love that later once I get. Well, yeah, it's really interesting field because you get to see that there's more to you know classical machine learning, or there's more to machine learning as a whole than just training on X data predicting a Y value, that kind of thing. But if you like the link, I would be sure to do that. Let me see if I can find it. So yeah, so at least it's good to see that the other people on the call are interested. So okay. All right. So my YouTube is loading. Once it's fully loaded, I'll be sure to drop the link. But yeah, you guys should also contribute to this conversation. So I think I'll pick someone else. I just ask them. Yeah. Hi, Michael. Akindele Michael. Talk to us. Is there anything don't know me, anything the AI space is experiencing? Michael, I said. Michael, I'm shooting my shot. Yeah, you're leaving me already. Okay. Okay, since our speaker is back. Chema, are you there? Hmm. Okay. Since she isn't. If you get to contribute to conversation, our organizer. If you're not there,
Hello, everybody.
okay. Hi, Chema.
Good afternoon.
Good afternoon. I'll
I
be handling.
apologize for the. Can I share my screen?
Of course, let me stop sharing mine.
I apologize for the um delay in coming on this call. So there was a misinformation. Your LinkedIn post says 29th. So it says Friday 29th, so I thought it was 29th. Yeah. Um can you hear me? Yes. All right. Thank you. So again, apologies um for that delay. So let me share my screen. Just go ahead and share my screen. Um, you can see my screen, right? So I can't see anybody. If you can just unmute and just reply because
Yeah, I
once
can see
I
it.
share that. Okay. Thank you. Okay. Let me just go back to that. Okay. So I have about 2 slides and I don't want to mix them up. So apologies again for just joining. So to briefly introduce myself before going into what we're going to look at today, in genomics ML, accuracy is not equal to equity. And this is a part 2 because in December, I joined Machine Learning Lagos Community Day. I think that was the first community day Machine Lagos community had, and I was opportune to join that as a speaker, and it was a very enlightening event. Again, thank you so much to the organizers of this webinar for the invite. So, brief introduction. Choma, a science girl, obviously. I'm a bioinformatics PhD researcher, currently rounding up my PhD. I'm also involved in global training and capacity building initiatives in Africa and most parts of Europe. I'm also a bio curator. So BSc, MSc, Biochemistry, Molecular Biology, Covenant University, and currently doing a PhD in Bioinformatics, Population Genetics. After graduation, I faced lots of rejection. So I'm briefly sharing this as a preamble to what I'm going to talk about today. Maybe it would motivate someone here to take whatever they are doing seriously, because it could lead you to some of the best opportunities of your life. So I know another thing I know is that people like to listen to gist and they like to hear gist. So instead of moving deep into the technical part, just to, say some things that would set a foundation to what I'm about to, discuss today. So shortly after graduation from school, one would think that, oh, first class degree, you get a job and all of that. But I was met with tons of rejections, and I like to call them redirections and reality check. And it was going on for, I think, about 2 years, rejections back to back. And this is just a snippet of the rejections. Trust me, there are quite a number. And some of the most painful, I had to remove it from this, presentation. So, but like what I like to ask is, are you hungry? Because for a hungry person, they don't stop at no, they don't stop at any negative feedback. They keep going until they achieve or hit what they want to, what they envision. So applying for opportunities, applying for training opportunities to be able to supplement what I was learning in school, because I'm currently doing my PhD in Covenant University, Nigeria, and obviously we know that some of the resources, or doing a PhD or postgrad degree in an underrepresented or low resource setting like Nigeria can be quite a lot, because sometimes when you need some resources, it's sometimes out of reach or hard to reach. So I had to apply for trainings in machine learning, computational biology, statistical genetics, to be able to, supplement what I was learning. And I did this by joining webinars and all of that. And from there, so this was actually a training that I attended. And one part of what I do is that I like to travel. Sometimes I get funded by initiatives in bioinformatics and computational biology to, travel and train people on what I know and what I'm learning. So this particular image by the right is where sometime last year, when I traveled to Uganda to train people on how to use my PhD tool, the tool I developed during my PhD, and it forms the basics of what we're going to look at today. And this picture also shows I was teaching them how to use in another training, how to use some codes related to logistic and linear regression. And the many pictures are when I was presenting and taking a picture in front of the screen. So shortly after that, I applied for some scholarships to get. So the bottom line is, I just kept applying for opportunities and scholarships to be able to, supplement my knowledge, because funds were a bit hard to reach then, and I didn't. You see some opportunities and they will tell you it's $1,000 and above, and you ask yourself, earning that I want to spend this kind of amount to learn what I want to learn. You have to eat now before you learn, because if you don't eat, what you're learning will not enter. So I learn and learn side by side. So I got to know about outreaching, and I applied for outreach. Outreach is a an initiative that helps individuals from underrepresented parts of the world who are, who face some technical challenges in their country. So they specifically target, women, men in some percentage from resources. So apply and you do an internship with them for 3 months, and they pay you. With that, you're able to get international international experience, and you're able to learn more about open source. It's basically about open source. So it's one of the most sought after open source program, and than God, and I as a mentor. So one thing about me is I'm a very delusional person. I love to dream big. I dream way outside my, how do I explain it? Way outside my league, because I don't know, I'm just a dreamer. So you see opportunities like applying when, okay, let's say someone sees two opportunities, or let's say I see two opportunities, one for machine learning intern or computational biologist, and I see one for senior machine learning engineer. I'm going for the senior machine learning engineer. Do I have the skills? No, but I would apply regardless. The worst they can say is no, right? And I learn and I apply again. But it doesn't mean that you shouldn't cut your coat according to your size. Your size, don't be like me. So this slide shows my first steps into open source, and after my outreach internship, blog posts, myself along with Esther and Peace, we were the three Nigerians that were that interned with Outreachy Bioconductor community via Outreachy, and our blog post was published on the official Bioconductor website. So one thing I would like to say is that you have to do good work in public. When you do your work in public, you are actually able to get opportunities and results. One thing I would always tell people I mentor and my students is that I wish I had started posting. I wish I had started sharing my journey on LinkedIn and online early, like much more early, because the opportunities I started getting when I started sharing it much later, I needed them when I was getting those rejections. So whatever you do, if you're learning, you don't have to be a pro in what you're doing. That is even how you become a pro. So if you're learning, whatever you're doing, is always good to share in public. Like, try and share what you're learning. It helps you learn better. So I started doing some mini projects. There was a project that a community called for, and they asked that if you get into the project, they give you a scholarship. And projects everybody did with GitHub, I went first for that, design a sweet interest cover. I designed this myself with Microsoft Word actually. Then I hadn't learned about Canva. Well, I think I knew about Canva, but I decided to use Microsoft Word for this, and I got accepted. I think out of we were about 40 that got the scholarship. So from there, from posting on LinkedIn, from sharing my journey on LinkedIn, I started sharing my journey on LinkedIn in 2024, and in that's 2024 October, November, December, and in 2025 I got invited to up to 17 talks from different organizations. I did not apply for most of them. They just found me through LinkedIn, and they're like, hey, I need you to come and talk to my community about this thing that you've been posting. And that was it. So I also got recognized by L'Oreal UNESCO Science Award for my PhD research, and basically my PhD on improving risk prediction for African populations, because there are actually risk prediction tools here, but when you apply it to African populations, it fails terribly. So one thing I try to debunk in my PhD research is that researchers, ML practitioners, that's machine learning practitioners, should not just focus on accuracy. You should look further down the line, like equity. This model that you say is 0.08 has 0.8 area under the receiver operator curve. When you apply it to Africans or whatever population you're studying, how does it give you the same level of performance? If it doesn't, that means your work hasn't ended. You have to keep going until you have achieved that. So that's what I advocate for. So I also got to chat with a journalist, a Vanguard journalist, Mr. Efosa. Quite an interesting chat on the tool I built for my PhD. So my vision is to increase representation in genomics and bioinformatics in Africa, and everything that I have done from 2021 to date has been around that. Capacity building, my PhD topic, every talk I have been invited to, and every talk I accept has been around that. So I in 2021, I left a 10-year career journey in biochemistry to start bioinformatics. Many people were scared for me. I was even scared for myself, but um, I'm very blessed, and I think I wouldn't call it lucky because it took 9 years of hard work, tears, and lots of rejections to get here. And this is just the least of it, because there's so much more in the pipeline. By God's grace, we are going to get there. So these are some of the projects that I have been involved in and I am currently working on. Project DigiBio 1000 is a project that I for to train 1,000 beginners and early career researchers in bioinformatics. And I'm currently on number 864. And my PhD research on polygenic risk scores, which we'll be looking at. And last year, I was appointed as the president of the International Society for Computational Biology, which is a community for helping computational biologists or students who are interested in life science students or machine learning students or comp science students who are interested in computational biology or bioinformatics, teaching them, equipping them with skills through webinars and workshops and so many other initiatives that we're doing. The link to our community is this. So let us move to what we are what supposed to look at today. I'll just share that because it's a different slide and I didn't have the time to merge the 2 slides. So let me just stop sharing that and share this slide. So I think this is it. No, this is not it. Let me see my screen. I wonder why this is not showing though. Please let me know if you can see my slide currently. Anybody hear me?
Okay, it's someone says it's coming up on their end. I can't see it on mine though, but I guess others
Okay,
can.
let me try. Let me try that again. Can you see my screen now? My slide actually.
Not yet.
I can't see it.
Okay, so I think it's still loading because on my end it's showing sharing. So let me
All right, it's come up.
okay. Is it on presentation mode or slideshow? Is it on slideshow?
I don't know. Stopped
It's
sharing
gone up
again.
my end. It's not showing again.
It stopped again. So
Yeah.
so what I'll do is that maybe I'll leave the meeting and join. I don't know why that is happening. So let me try to leave and join again. I don't know why that is happening. Maybe I don't have to leave after all. Can you see my screen? Is it in slideshow?
Yes, it is.
It's on slideshow now.
Yeah, it's full screen, but I can see your task. Your taskbar is covering it a bit.
Okay. No, that is not supposed to happen. Is my taskbar still covering it?
No.
Yeah.
Okay. So, looking at the topic for today, which is in genomics ML, accuracy is not equal to equity. So we'll look at that shortly. But before we start, I would like to know who is in the room with me. Maybe the moderator can tell me the an overall statistics of the people here. Are they just CS students, that's computer science students, or just machine learning students, or a mix of machine learning and biology?
Okay.
I just want to know how I would break things down.
Sure, no problem. Okay, so first off, it seems the screen is down again.
Okay.
And second of all, to answer your second question, your question, we have a combination of, students, working professionals, and someone in the chat just said they're microbiology students. So yeah, we
Okay.
do have a combination of people from different backgrounds, from students, researchers, and people of all different levels. Right, a very diverse community.
Okay. Okay. Very good. Thank you. So what I'll do, I think the solution now is just to leave it this way, because if the moment I put it on, presenter mode, it just starts showing something else. So I think this might be the solution for now. Or let me try something again. And I really apologize for this. This is strange. I don't usually experience this. So I wonder why. Maybe I should put it at the bottom. So let me know if anything is disrupting. Is it okay now?
I can't see the screen personally.
You
Anyone?
can't?
Yeah, I can't. I
Oh
can't,
my
can't see it again.
goodness. I'll just leave it the way I mentioned
Yeah.
earlier. I'll just put it this way. I think
All right.
you would still be able to see it like this, not just
Yeah, I can see it now.
I think we should just permit me to just use it this way. Thank you. So should be better. So again, this is the outline. I'll be, talking about the basics. What I mean by genomics for ML is, what do you mean genomics for ML? Is ML not ML? Is genomics not genomics? No, in my world and in my community, it's not like that. We'll be looking at the gaps and a thinking frame, the tools that you can use, and then a thinking framework that is applicable to solving this challenge. So translating genomics into ML terms, because I like to, instead of assume, I had to make that, ask that question, what kind of people we have here, or in what fields. So your genome is about a 3 billion letter string, and we call that 3 billion letter string nucleotides in biology. So there are about 4 of them that repeat each other, and they are arranged in a certain sequence in your genome. The way it's arranged determines what kind of proteins your body make. It determines what you look like. It determines it helps us to know that, oh, your mom is dark, your dad is light skinned, and you inherited your mom's skin tone, that's chocolate or caramel skin tone. So this is what they call the genetic code of life, and our genome consists of about 3 billion of these. And the interesting part is that we are all different at different positions. One person's A is another person's G. And these differences is what we call variants. And a specific type of variant is called SNP, SNP for short. The full meaning is single nucleotide polymorphisms. We won't be looking into that, don't worry. I just wanted to lay a foundation. So in machine learning, we take the features as these variants, and they are often about maybe millions or 100,000 of them in our genome. So, and in some machine learning models, the labels has to do with the phenotypes. Either is a trait, maybe height, or a disease, maybe diabetes. Each row is the person's genotype. Now, looking at this matrix here, I hope my cursor is, yeah. So looking at this matrix here, the genotype matrix, by the rows is each person's genotype, and you can see the variants and the features. Light meaning double A's, light blue meaning double A's, light green meaning AG, and deep green meaning double G's. This is just to, paint like a mental model of what I'm trying to introduce. So what machine learning practitioners do with this kind of data set today? Now, all this, what I just explained, they are deposited in databases. We have different databases. We have the Cancer Genome Atlas, TCGA, we have GWAS catalog, we have PGS catalog. There are different databases. There's Gene Expression Omnibus for getting biological data sets. So machine learning practitioners go to these databases to pull let me not just say machine learning practitioners, because that's too niche. Computational biologists, anyone interested, goes into these databases, and they use this data for either for risk prediction, that's to predict risks from the person's genotype, maybe using penalized regression, XGBoost, deep nets, deep neural networks, polygenic risk scores. They also use this data to do something we call variant effect prediction. That's to score whether a variant is either damaging. Sometimes when someone has a mutation in their gene, biology helps us understand that this mutation, sometimes it might not be so bad. It's just there. There are some mutations that are beneficial. There are some mutations that, instead of it to be bad, is actually beneficial to the person, right? So this variant effect prediction, there are some people who actually do these things. They have fun studying these things. They study whether a variant, in biology, we call it missense. We call it, there's nonsense mutation, there's point mutation, there's silent mutation. So another thing they use this data for is also for imputation. Quite a number of things: phenotype prediction, using genotype and clinical features to predict whether a person is likely to get diabetes or cardiovascular diseases or not. There's also something we call functional genomics, which is one area I'm also into, to predict gene expression, regulatory activities. There's another one called population genetics. This one deals with ancestry. There are different interesting parts of what we can use biological data for. And this is just, just a handful of it. There are so many applications. There's also, this is just on genomics. There's microbiome, there's agriculture. There are different aspects, but I'll be looking at genomics today. Now, when we say explainability, what do we mean? Oftentimes, when I interact with students, I get to understand that, especially computer science students. I love to interact with computer science students a lot because I don't know, it's like when I discuss with them, it makes me feel like, it makes me understand how like people from other aspects think, because I'm coming from biochemistry. I learned computers, I learned some programming and some coding foundations from a biochemistry angle. I had to start afresh to learn all those operating system and all of that. I like to interact with computer science students because they help me think, or they give me the perspective of how comp science think about models, and it also helps me in the way I translate that into what I do. And I like to ask some questions when computer science students or some machine learning practitioners build models. What I see on Kaggle, for example, is they just put these models and that is it. Maybe they tell us about area under the receiver operator curve, that's AUC, or they tell us about R square, or they tell us about precision, F1 score, and all of that. But there need to be a way for someone to go further. We shouldn't just stop there as machine learning practitioners or researchers. We shouldn't just stop there. You should actually go a step further to ask, why did your model even predict this? Why is it giving this specific accuracy? What kind of features drove this prediction, and how did it drive this prediction? This is one way to discovery. This is one way to trust and safety, especially in this era of AI. So that is what explainable AI is about. Is a set of methods that answers the why of your model. But let's look at something. There is a there's a problem, or a let me call it a problem. It's a problem, but I'm looking for a stronger word for that because it's more than a problem right now in genomics. But this is a data from GWAS. European populations, the representation of European data sets in GWAS. GWAS is Genome-Wide Association Study, is one of the data sets we use for computational biology research in polygenic risk scores. 86% of the data sets in the GWAS from GWAS is from Europeans, and just about 1% is from Africans. And now this is what we call lack of diversity in genomics, and African populations are one of the most diverse in the world. And we are saying that we want precision medicine. There's no way precision medicine can be precise without the most diverse populations data inside it. So the image to the left is the representation of the data sets. The image to the right is the accuracy. So it's not far-fetched that when you use data sets from a specific population, that there is more from that population. When you build the model, obviously it will perform more for that population, right? Now look at Africans in this plot, in this graph. Is the purple looking bag. So you see that Africans it's very low in terms of accuracy, and this is a problem, and this is one of the motivations that drove my PhD research. Now, the solution, one would say that the solution is to improve the data sets, right? Because African data sets currently is very limited in databases. One would say is to improve data sets, more research should be done in Africa for Africans by Africans, Yes, we've heard all of that, and it's very important. I'm not undermining that. But we, as machine learning researchers and computational biologists, so to speak, can actually do better. So we're not in wet lab. So the wet lab folks would help in improving this data representation policy folks would improve policy, equity, and all of that. We have a role to play as machine learning researchers and practitioners. And what is this role? When we build models, when we design tools, we should try and consider the uniqueness of our genetic architecture. Our genetic architecture is different, very different from other populations. And there are some things we'll look at in this talk, and I think that's the next thing in this slide. Yes. So there are some certain assumptions that current prediction tools make, and they hard code the quality control thresholds. QC stands for quality control. They hard code the data formats. They assume some things like linkage disequilibrium structure and the likes. And so briefly explaining linkage disequilibrium. Linkage disequilibrium means the non-random association in the alleles. I think I'm going too technical in terms of biology. I'm just trying to, let me use a smaller let me try to bring it home to our comp science folks here. So there are the arrangement of our alleles or letters, since I said with letters in our genomes, is the way it associates with each other or correlates with each other is different from other populations. And now there are tools that exist that, consider these arrangements, and they integrate it into the tool such that when you are using it on African data, it considers this uniqueness of our African genome. There is a call for machine learning practitioners and researchers to, when designing tools, consider these things. There's a host of them. Apart from linkage disequilibrium, there's something we call allele frequency. There's a number of them. So it's good that we equip ourselves with knowledge about this, because you can't solve what you don't understand. You have to understand the core of the problem. That's for those who are actually interested in doing research or doing groundbreaking work in health or genomics. So there are reasons why African genomic data are underrepresented, and it shouldn't be new to most of us, but just to reiterate it. Funding is part of it, infrastructure is part of it, capacity and training. There is a limited pipeline of bioinformaticians and computational biologists and ML researchers who actually know the urgency of this problem we currently face, inequity in current prediction models and other models. There's also the thing about the diversity itself. There's also the thing about trust and governance. Different reasons why African genomic data are underrepresented. One of us has to be the solutions. Not just one of us. All of us can be solutions, but we have to start. So there's a thinking framework I would like to propose for genomic ML models. 5 questions, but that you need to ask when you want to start a project, if you're working in health or genomics, and don't stop until each is answered. The first is performance. We are asking questions like, does the model work at all? Right? And that's where most, or let me not use most to generalize, where some machine learning researchers stop. Once they get their accuracy, they are done with the model. You should take it a step further and ask, who does it fail? Who does it work for? Acquire these data sets, test it in the population you're interested in, and find out who this accuracy now, is it the same? Does it give the same level of accuracy in Africans? Does it give the same level of accuracy in East Asians or whatever population you're interested in? Even within Africa, there is diversity. We have sub-Saharan Africa, like West, East, Central, and then North Africa. There was a study that they built a model in Ghana, or I think the model was built in Nigeria, either one of them, and it was tested in Ghana, and the accuracy dropped. So even within Africa, there is diversity. So you can actually pick a side of a region in Africa if you have access to the data set and work on it if you're interested. You should be interested. That's the point of this talk, right? That's if you're into health and genomics research. And then the next thing after population gap is explanation. You need to be able to explain why the model fails. And we'll be looking at that shortly. Then you also need to explain the biology. Does the signal make sense? So that's where collaboration comes. If you feel like I'm just a computer science, I'm not interested in biology. Biology is just too long and all of that. It doesn't mean you shouldn't learn. You can learn a bit of biology and then collaborate with biochemists, microbiologists, people who are actually in the biology field to be able to understand what your model means and then validates, because after you test, say that, oh, this accuracy is this, I'm able to explain this is this, you have to also test in an external cohort of everything you have done so far. So accuracy is only step one. It is not the finish line. So the next is I just want to introduce you to some tools. I think you should even know some of these tools, right? Some of us here should know some of these tools. There's Shapely. So Shapely is when normally in machine learning, when you're done building your model, Shapely gives you information about the variant IDs. So you can do per group Shapely, you can run Shapely separately per ancestry to know which of the variants drive the predictions in each of the population. There's Lime, there's Deep Lift, but I'm more familiar with Sharp. Then there are also some pathways and some databases that you can, don't worry, I think I'll share this slide with you so that you can have access to these resources. There are some databases that actually turn those variant IDs into biology. You need to understand what gene those variants are in, what kind of protein it changes, and how often it appears in populations. And there are some databases for, there's GenomeAD, there's Variant Effect Predictor, that's VEP, there's ANOVA, there's CleanVar, there's GWAS Catalog, there's database for genotype and phenotype, that's DB-SNP. So after you look at your genes, the variants involved in your model, whatever model you've built, there's something we call biological pathways. So biological pathways basically means that there's a way those variants or those genes cluster and they drive a chain of reactions, interacting with in biochemistry, we call it mechanism of reaction. How is this change occurring? What pathway? Is it the prostate cancer pathway? If it's the prostate cancer pathway, how does it move from what protein to what protein, this kinase, this enzyme, how does everything come together? What is the big picture? So this is what it tells you. So now you see that there's actually a lot of beautiful stories to tell after your ML model. And if all these things seems like a lot of work to you, you can actually collaborate with a computational biologist or someone who is into biology to help you out with these things, or actually go and read some resources, which is here. I think I have them here for you to learn how to do these things. On YouTube, there are resources on how to learn how to apply these things. There's the Kyoto Encyclopedia pathway, there's KEGG, there's Reactome, there's String. You can do this and publish and present your work in conferences and tell people about what you're interested in. So this is you don't need a PhD to be able to do this. You trust me, you do not need a PhD. If I can turn back the hands of time, then I thought I needed a PhD, but if I can turn back the hands of time, I would have done trainings first before doing a PhD instead of the other way around, so that I would know the level of knowledge I need to or the level of skill gap I lacked, or the skill gap to fill before I started my PhD program. But anyways, we can't turn back the hands of time. But yeah, just to emphasize that you don't need a PhD, you just need enough biological understanding, because if you're interested in health research, if you're interested in genomics, you cannot solve what you don't understand. You have to understand the biology itself to an extent before you can solve problems related. And I would also recommend Andrew Ng's courses and YouTube. He has some courses on Coursera and YouTube videos as well. There's StatQuest, a host of them to learn from. Thank you so much. And if you have questions, I don't know how it goes, but moderator, if there's questions, if anyone has any questions, I can answer. Yeah.
Thank you so much, Shema. Please, if you have a question, you can just raise your hand and go ahead.
Jesus.
Okay. It seems that there are no questions. Any questions, anyone? All right. Please go ahead. Heidi, you can go ahead with your question.
Okay. Thank you very much. I don't know if anyone can hear me.
Yes, I
Loud
hear
and
you.
Thank you, Chairman, for the last presentation. I was really impressed. I myself am interested in, I don't know if it's computational biology now, but still something very similar, which is in predicting like the risk of cardiovascular diseases like hypertension and stroke among African population, and then using it as to just inform preventive medicine. So now I know you mentioned something about being president of the International Society of Computational Biology, if I'm correct. I just wanted to ask,
Yeah.
what kind of people, what kind of roles is that society open to? And say if somebody
Um,
is interested in joining, how can they go?
your audio was breaking at some point. If you could repeat your question, please.
Okay. So I said for the International Society of Computational Biology, if somebody wants to join, what kind of roles or people are looking for and what criteria does it take to join? Like how do you join?
I'm not sure I got that. Can anybody help me to repeat what he said?
Hi,
Sorry.
Heidi. Can you type out your question? Yeah, audio is a bit choppy. I couldn't make it out too. So just type out your question in the chat. Thank you. And she'll be sure to answer. So Chioma David is asking the list of things you have to check out. Are they going to be in your slides, or you made a list of things that you mentioned a list of things for us to check out after the meeting. I wanted to ask, are those things in your slide? In your slide, right?
The is it resources?
Yes, resources.
Yeah, it's here. It's on the
Right.
slide actually. The last slide.
So she's going to share the slides, and you have access to all
Yeah,
of
I'll
them.
share the slides with you. So I'm
Okay, so
still waiting
we're
for
waiting
him to
for
type. Has he
yeah,
typed?
he hasn't typed it on yet. So we're waiting for Kyle to type out his question. If you have any other question, please feel free to raise your hand or type it out in the chat. Okay, it seems he sent it.
Okay. Okay, so I'm interested in the use of ML to predict the risk of cardiovascular diseases in African population. Could that count? Would that count as computational biology? Also for intentional. Come on, Jen. Okay, so computational biology is a field of, some people will say it's a field of computer science, some people will say it's a field of biology, but it is an interdisciplinary field that deals with application of computational techniques, algorithms, tools to solve biological questions or problems. And sometimes it can also involve development of applications to solve, and development of tools to solve biological tools or questions or problems. So biological questions meaning it encompasses cardiovascular, maternal, women's health, men's health, health, neuroscience, and so on. So yes, cardiovascular diseases, research in cardiovascular diseases counts as computational biology. The link to join International Society of Computational Biology is actually on the slide. It's the barcode was there, so I would add that slide to this one that I'm going to share with the moderators or the organizers of this course, and they'll get that across to you. I hope that's okay. Thank you.
All right. That seems like all our questions.
All right. Thank you.
Thank you so much for the session, Chioma. I really learned a lot. And it was great to have you again.
Yeah. Thank you so much.
You're welcome. Okay, so you can share your screen now, Chioma.
Yeah. Okay. If there's no other thing to do here, I'll have to jump into another meeting. So
All right.
is that
No
okay?
problem. That's totally fine.
All right.
Thank you for
Thank you.
joining us.
Bye. Thank you so much for having me. Bye.
Okay, everyone. I hope everyone enjoyed the session. Just a little bit small before we leave. We will be, so our upcoming events are going to be having the June office hours, technical session, and the MLA Girls Grad School series. So if you are interested in grad school, I think I've dropped the link in the group chat before. So if you're interested in grad school, you just want to, get some information, Machine Learning Lagos does a grad school series. It's a 4-week program, so it's going to kick off on June 7th. And if you are interested, you can also go back to last year's grad school series. It's on our YouTube channel. So let me just drop the registration link once again on the group chat. No, not on the group chat, on the call. So I'm going to drop the link for that here. Please register if you haven't already. And also, yes, we'd also like your feedback, on this session. If there's anything, that you want us to do differently or anything we can improve on, I've also sent the link for that on the chat. So thank you so much. And also there were some people that said they wanted the link to the documentary I was talking about. So I've also sent that to the chat too. So be sure to, check that out. Thank you so much for joining us this session, and I hope you have an amazing rest of your day. Thank you very much. Have a good day, everyone. Yes, of course, the link for the recorded session will be provided. It's, immediately, all our sessions are recorded and it's going to be on the YouTube channel. So you can just go there and all resources will be shared too. So the slides, the speaker slides will be shared, and the link to the session is also going to be shared. So you can check that out on our YouTube, or since you're on the group chat, I'm sure you'll be able to see. It's going to be announced, like we'll post it. Sure. You just hang on tight. Thank you. So I think I've given some time for people to click on the links and check them out. So thank you very much, everyone. Have a good day. Very nice session. It was lovely to have you.